This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 616, recorded on May 20th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 61, which is 16. And let me see, I have to stand up to look out the window over my tent. Yes, it's currently bright and sunny.
Wow, it's the same temperature as it is here, and it matches our episode number, which is cool. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It's 80 degrees, mostly cloudy, kind of humid. I'm just as happy to be inside today. This will do just fine. It's headed for 95.
You must have the AC on already, right?
Yes. Oh, yeah. The AC is on. But, actually just recently, there were times as recently as a week ago when we had the house all open. Very nice.
I just turned the heat off a couple of days ago. I get a couple of weeks with windows open, which I really like, and then they'll get closed for the AC, which I don't like. But I'm outnumbered here. There are 3 people besides me, and they all want the AC on.
Going to go down, according to this, it's by next Monday, it's supposed to go down to 83 with some thunderstorms. But basically, we're strolling our way towards real summer here.
If you like what we do here on TWiV and our other podcasts, consider supporting us, microbe.tv/contribute. All right, we are going to mainly do email, but we have a couple of stories, which I'm going to say are going to take an hour probably. Usually what happens.
By the time we're done, yeah.
Because you can't shut scientists up.
Can't shut them up, and that's fine. That's what we do. So we're going to start with Kathy with this lovely picture of a wave.
Right.
Reminds me of that Japanese etching, right? By the
the tsunami.
Totally. Yeah. So this is an article in Science by Jeffrey Brainerd, and thanks to my brother Bill for sending me this. He has more time to read science at the moment than I do. So this is the Hokusai wave, showing how scientists are being inundated by papers about COVID-19 and SARS-CoV-2. And it's just full of interesting statistics. There's more than 4,000 new papers last week. This was published May 13th, so base it on that.
Is that like published or preprint?
It just says more than 4,000 new scientific papers. So, yeah. But published since January, it's greater than 23,000, and it's doubling every 20 days. As of April 14th, 80% of the papers had been published in referee journals, probably due to accelerated publication.
Wow.
Currently, 80% are open access, but that could decrease to 50% by June 1st based on trends. And in other words, about 50% of the papers at that point would be behind paywalls. And part of this, or the main thrust of this article, is to talk about new search tools that have been really kind of little known outside of research groups, and scientists haven't heard of them. They use sort of literature mining algorithms. They aren't necessarily user-friendly, and many scientists are skeptical that these can tell them what they want to know, which is, what is the quality of the work? And so people tend to oversell and put up papers with data that don't support their conclusions. And so that can be a mess for this. And so there's 2 basic strategies. One is creating easily accessible paper collections that are somehow curated. And the second is building automated search tools. So, and those search tools.
Actually, the third is to listen to TWiV. But
well, yeah. Yeah. Or the old fashioned way, just on your own, just kind of scan the literature. But
I've read every single one of these in detail.
Right.
You're retired, man.
So, so this search tools thing uses artificial intelligence. So they mentioned 2 data sets. One is called Cord19, and there's a link to that. It has more than 59,000 published articles and preprints, including studies of coronaviruses going back to the 1950s. But evidently it's flawed. Some of the papers in there seem to lack mention of any of the search terms, and a third of them don't have the full text. And then there's a second data set that's being set up by Johns Hopkins. So again, in addition to doing the dashboard, now they're doing this data set. It's led by Kate Grabowski, an infectious disease epidemiologist in the School of Public Health there. And it's called the 2019 Novel Coronavirus Research Compendium. It's curated by 40 scientists, and so far it's 80 papers with summaries. So those are the two data sets. And then in terms of data mining, this thing, one is called AI-powered literature review. This work is far from being automated, so the algorithms don't always extract the right data. So at the moment, they're repurposing medical students and other volunteers who are idled by the pandemic and having them check the things that turn up against the manuscripts for accuracy and so forth. Then there's a user-focused tool called SciSight, SCISIGHT, and that can be used to mine that first COVID-19 data set that I mentioned. And it was unveiled last week and automatically provides suggestions of papers containing related themes. So these tools are not all that well known to many scientists, including me, I will admit. So a half dozen scientists contacted said they sounded promising, but they hadn't heard of them. And others added that they didn't have time to try them. Okay, right. I get that. And then one person, a data scientist, Jevin West from University of Washington in Seattle, said, yeah, it's hard. It's like bringing a new scalpel into the emergency room and saying, here, try this. It's actually better. And, of course, the ER docs are like, I don't have time for that. So that's how that is. So back to this Johns Hopkins database, it's focused on studies in humans, and the target audience is healthcare workers and policymakers, as well as researchers. It has excluded most of the articles that are originally considered because they contain commentaries, protocols, poor quality modeling studies, or no original findings. So they are, sounds like they are being a pretty good filter. And, as one other researcher points out, there's so much going up, and this faster dissemination might be at the expense of research quality. That's something that Nels and Vincent captured with their title Rough Drafts of Science on a TWiV episode a couple of weeks ago. So, yeah, that's the article.
So this is all good, I guess, because, some people are thinking, let's see how we can improve things. But I'm not sure that, most scientists want this. They have their ways of doing things. And until it's obvious that this is great, they may not use it. So I use a novel tool, which I call reading. I look at the title and the title can eliminate a good deal right there. Then, if it says mutation that leads to increased transmission, that's the end of that. And then I look at the abstract, and if it makes it past there, then it's TWiVable. I don't feel overwhelmed by this, because you can sort through a lot of stuff as these machine things are, like commentaries, protocols, poor quality modeling studies. I don't know. I mean, I would look at this stuff at some point to see if it's useful, but I don't feel overwhelmed, and I'm doing a lot of reading. But that's what I do for a living. I love it, right?
Mm-hmm. Mm-hmm.
My method is that you guys send me stuff.
Yeah, people send us stuff here too,
and I read it.
Well, and you get some from your Brighton collaboration and stuff.
Yeah, the Brighton collaboration has been great. There's one of the members of the Brighton collaboration, a gentleman who actually has a very interesting career as a vaccine researcher. He's French, but stationed in Singapore, and he must read like a madman, because I'm constantly getting stuff from him, and it's been very good, very nicely filtered.
Yeah, it's true that our listeners send a lot of stuff to us. That's a good filter. And various people are always sending me stuff, but I also scan and find what I'm interested in. I'm not sure at this point that I would trust any curated collection that's not going to, I mean, I have specific interests too, and maybe they're not going to be represented there. So unless you put some time into seeing if these are useful for you, and that's not it's not an available commodity right now anyway. But, that's how things get developed, by someone thinking, let's try this, and it either works or doesn't. Mm-hmm. It's like a cell phone, right? Let's make a cell phone without a keyboard. Mm-hmm. And it works.
What a revolution.
And people say at the beginning, I can't type on glass, but turns out you can. Mm-hmm. But who would have known unless you tried it? So I'm all for trying stuff. It's good. Well, thank you for summarizing that. I would never have looked at that, actually. But I do have this general concern about people. I think someone quoted here said they're putting up papers where the conclusions are not supported by the data. That always happens in a situation like this. You get a furious rush to publish. I mean, I'm always amazed at what becomes the least publishable unit, the LPU, especially for a high-profile journal. I'm like, really? Mm-hmm. And I was thinking this morning, I was trained to say you have a paper when you have a mechanism, not just an observation. This goes up under these conditions. I wasn't trained to publish that, but now it gets published all the time, right? Mm-hmm. And I guess one of the philosophies is you put it out there and let people look at it, but then it seems to me that shouldn't be in a high-profile journal. But the journals are trying to make a living and stay alive and everything, so this is all tied together. Mm-hmm. Yeah. I mean, if I publish a paper without a mechanism, I get criticized when it's time for grant review. This individual has not published any mechanistic papers in 2 years. I've had that. So why are all these non-mechanistic papers getting published? Anyway, it's the way I look at it. I agree that I think mechanism is what drives science, but there's obviously a lot of observation out there. Mm-hmm. Rich, how do the different vaccine platforms work?
Wow. Funny you should ask. I don't know where I came across this. I should say another one of my literature sources is Ed Niles. He feeds me stuff all the time, and I think this may have come from him. This is now you could even call it dated. It's 3 weeks old. 28th of April, a news feature in Nature titled "The Race for a Coronavirus Vaccines: A Graphical Guide," and this is a really, I think, a really nice, easy to read, graphical, sort of diagrammatic guide of how vaccines work in general and how the various vaccine platforms that we're hearing about work more specifically. And I think this is a really nice summary for people who are interested in this. And we're going to be going, a couple of weeks ago, we discussed a viral vectored vaccine, the one being created in the chimp adeno vector by the Jenner Institute. And
that was just a week ago.
Oh, was that just a week ago?
Yes.
And today we're going to talk more about an RNA vaccine. And I think this graphic is a nice background for all of these things to show you how they work. And I have a bunch of immunology questions. And no offense to you guys, but on some episode where Brianne is around and we're discussing a vaccine, I want to dump all my immunology questions on her. A lot of those are at least outlined in this graphic, but I have a lot more questions that I want to ask her.
So let me just be pedantic here. The virus uses its spike protein to lock onto receptors.
Yes.
So please don't do that, folks. The virus doesn't use it. It happens to bind the spike. It doesn't use it. That is giving it an active aspect, which isn't correct.
But it could be, I know you're being pedantic, but it could be that the person has written it as the virus uses its surface spike protein with the result that it locks onto. So the word "to" is what kind of gets people in trouble. when the virus uses something too. So
actually, it sounds to me like Vincent's taking exception to the word use.
Use is what I don't like.
Oh,
use
yeah.
virus
Yeah.
is what I don't want.
Yeah.
You should say virus binds ACE2 via the spike or something like that. There are many ways you can do that.
Right.
But, I see the person who made this is not really thinking about this all the time, whereas I've been thinking about it for 80,000 years.
Wow.
And the other thing, I know I'm really old. The other thing that I noticed here is they call attenuated viruses weakened virus.
Yeah.
This is absolutely unacceptable. Weakened virus. This is a judgment word, weakened. Okay. Mm-hmm. It is an attenuated. Let's use a scientific term. I'm already getting excited here.
Overall, though, it is a very clear presentation of the various things. And I only had one question that I put into the notes, which is about the DNA-based vaccines. And I don't know if either of you guys know the answer. Of the 2 nucleic acid vaccines, there's DNA or RNA. And they show that for the DNA, you electroporate the DNA into cells, and electroporation creates pores, allowing the DNA to go in. How are you going to electroporate a vaccine?
I believe that there is an apparatus.
Yeah, there is a
that
gun.
effectively electroporates DNA into people.
So you put it on your skin and okay.
Something like that. Yeah.
Okay.
I think one of the names, it was called a gene gun. And there are 2 approaches. One was it would shock you while putting the DNA in, or another one was actually to put it in under high pressure air that had compressed air in it.
Right. Okay. Yeah.
It in. But yes, it will shock you.
I do remember the gene guns now, but I encountered them initially, I think, with plants. So
for the TWiVers out there, I imagine that one of the reasons that Kathy is asking about this is that in the laboratory, electroporation is a long-standing technique that involves sticking a solution of, say, bacteria and DNA in what we call a cuvette, which is just a small vessel with, in this case, electrodes lining the 2 sides of it, and then passing a current through it, fairly vigorous pulse over a short period of time. So, if that's your background, it's hard to imagine, like, what are they going to do? They're going to soak you in DNA in a big tank and run a current through the tank?
Yeah.
They'll put you in a Bacta tank. I don't know if you guys know what that is.
What?
It's a Star Wars thing. They put people in a Bacta tank to heal them. To tank So this picture of the nucleic acid vaccines, they show this RNA vaccine fusing at the surface, and I don't believe that's the way these are thought to get in. I think
no,
they're endocytosed.
That's another problem, and that I looked this up at one point as to there's a couple of different ways of packaging an RNA vaccine. The most common you see is what do they call them? Liquid. They're not micelles, and they aren't vesicles.
They just call them lipid nanoparticles.
Yeah, lipid nanoparticles. And the thing is that's not a bilayer. Okay, it's on the outside is effectively a half a leaflet for the aficionados, half a leaflet of lipid with some fairly disorganized lipid on the inside. And so that's not going to fuse with a membrane in the typical sense that a lipid bilayer does. I think you're right. They're taken up by endocytosis or something like that and dissolved. So yes, this has some, this is not perfect, but a nice graphical summary nevertheless.
So let me give a little suggestion to these journals who want to make these graphics. Run it by someone who has experience in the field and who has the time to look at it.
Right.
Because if people don't have time, they're going to look at it and say, yeah, this is fine, without looking at the details. I mean, someone from Scientific American sent me a graphic for PCR. I had to spend 30 minutes to make sure everything was right. And that's how you avoid stuff like this. And, not everyone's going to worry about the virus uses. Mm-hmm. So you need to get, someone who's going to, because, and in the end, it doesn't matter all that much. Actually, it does. I don't want people to think that the virus uses anything. Mm-hmm. Get the right reviewers. It's hard, I know.
Get somebody who writes a textbook where they read the chapters out loud to each other.
You could do that, or Kathy Spindler would be perfect. review all this stuff. She absolutely gets it, and Condit too would get it.
I don't know. I'm likely to write a book that's got a bunch of anthropomorphic stuff in it and all the wrong vocabulary and everything else.
It's all right. We'll call you out. We'll say, "Hey, what are you talking about here?" In a good way, though. In a good way. Now, with that as a background, I thought we would discuss briefly Moderna's phase 1 mRNA vaccine trial, which the news came out this week, and as Trina Suderos. I don't know if both of you remember her.
Wow, what's she doing?
Mm-hmm.
She is working for a healthcare organization, and she emails or texts me all the time, and she said, "This is science by press release."
Yeah, really.
It's a press release, no data. I hope we see the data at some point, but they did a phase 1 safety trial, and they released the results. And Rich, do you want to summarize it?
So it's the safety trial, and once again, you can find all of these on clinicaltrials.gov. Is a phase 1, so that's not many people, and just the primary outcome they're interested in is safety. Open label, which means that it's not double blind. They know what's going on. They know who gets what. As a matter of fact, this one isn't even placebo controlled. Dose ranging study of the safety and immunogenicity of the 2019 nCoV vaccine mRNA 1273 in healthy adults. So what they're doing is they're giving 3 different age groups of healthy adults 3 different doses of this mRNA vaccine in both a first dose and a boost 29 days later. The doses are 25, 100, and 250 micrograms each. And as I said, they're boosting on day 29. The 3 age groups are 18 to 55. There are 15 in that group. 56 to 70, there are 10 in that group. And over 70, there are 10 in that group. So it's 105 people total. And the data, which I'm going to put in quotes here because we don't see any data. This is science by press release. And it's really frustrating. Reading this article, even, there are some inconsistencies in the way the reporting is done. And it just makes, I'm just itching to see a table. Not only that, but they're not finished analyzing all the data. This press release is because they have gotten some positive data on a subset of these individuals, and they're reporting on that.
This is a lipid nanoparticle encapsulated mRNA encoding a pre-fusion form of the spike. Pre-fusion, that means it has some amino acid changes so it does not get cleaved in the cell, which will rearrange it into a form that is probably not good for blocking antibodies. So you want to prevent that. And that's been shown with other viruses. So that's a clever way to get around that.
Right. And they just use genetics to put in appropriate changes in the amino acid sequence of the spike protein so that it stabilizes it in this prefusion form. And the technology here is that they have a messenger RNA that is actually, it's slightly modified. They use pseudouridine instead of uracil that has the effect of, I think, modestly stabilizing it, but mostly it's there because that an RNA with pseudouridine instead of uracil does not stimulate as much of an innate immune response. Okay, so that helps out with that. It helps it be more effective. So, and it's in, as we've said, a lipid nanoparticle. So
injected into the muscle, right?
Injected into the muscle. So cells will take this up, and it will be a messenger RNA in the cytoplasm, which will then be translated in the cytoplasm into the spike protein. And it is the protein that is made in the cytoplasm that you're making the immune response to. And there's all kinds of other stuff that impacts on exactly what is the profile of your immune response to this. And that's the conversation I want to have with Brianne. Because if I try and describe that now, I'll probably get most of it right, but it would be nice to have her on task to get it right. At any rate, what they measure as an outcome is whether or not you make antibodies to the protein. Not only that, but according to this press release, in a subset of patients, they measure whether the antibodies that are made will neutralize virus in a plaque assay. Good for them.
Mm-hmm. Yeah, good. But the data are not complete. They say all participants across all 3 dose levels seroconverted by day 15 after a single dose. So that means they made antibodies that bind the protein.
So that's great. That means that even at the lowest dose, 25 micrograms after a single dose, according to this, they seroconverted. Now, there's other text in here that indicates that, well, maybe the level of antibodies in the lowest dose, and I'm reading into this here, may not have been as robust as they would have liked. Okay, but others look great.
At day 43, which is 2 weeks following the second dose, the levels of binding antibodies for the 15 participants dosed at 25 micrograms were at levels seen in blood samples from people who have recovered from infection.
Now, day 43, that's going to be after the second dose. This to me is important because when we're thinking about deploying this as a vaccine, whether you can do this in one dose or whether you're going to need a booster is really significant.
At day 43, 10 participants at the 100 microgram level showed levels of binding antibodies that significantly exceeded the levels seen in convalescent sera. And, that significantly, they shouldn't say that because we're not seeing the data. Mm-hmm. So I don't know the statistics, so not fair.
Right.
Then they did neutralizing antibody tests, as Rich said. Levels of neutralizing antibodies at day 43. All right, so the vaccination elicited neutralizing antibodies in the first 4 participants in each of the 25 and 100 microgram dose level cohorts. And that's all they have data for that. And these, at day 43, neutralizing antibody levels were at or above levels generally seen in convalescent sera.
So that implies to me that after 2 doses of 25 micrograms, because they're talking about day 43 and they were boosted on day 29. Once again, we're reading between the lines because we can't see the data. But that implies to me that at 25 micrograms after 2 doses, you're seeing a level of neutralizing antibody that is equivalent to what you see in people recovered from the disease. That's great news.
Mm-hmm.
I mean, the question is, and this has been brought up in a couple of papers that have come out studying recovered patients and antibody levels in them. Is that enough to prevent infection? We don't know yet.
Right.
And the only way we will know is when they do their phase 2 and 3 to see if those levels are consistent with protection.
Right. And this is part of the conversation I want to have with Brianne, because I think I can say that we don't really understand what the correlates of protection are in this disease. All you're measuring here is neutralizing antibody. And with some cases, in some vaccines, neutralizing antibody alone is enough to give a protective immunization. But we're probably, I presume, because it's technically more difficult to do, so far we're ignoring entirely the other arm of the immune response, the cellular response. And for all we know, that's equally important. And we do know that depending on how you, what kind of a vaccine it is and how it's administered, you can get different levels, relative levels of the antibody response and the cellular response. I would hope, and once again, this is why I want to talk to Brianne. I would hope that somewhere along the line, they're looking at the cellular response in these people. But it must be that it's just technically a much more difficult thing to do.
Well, last week when we talked about the chimpanzee adenovirus, and I think it must have been from the actual animal trial, they had reported on the neutralizing antibodies, but they also reported on the cellular response by looking at interferon gamma. So that is something that they didn't put in the press release. Is that because it wasn't good results, or they haven't done it, or again, we don't have
interferon gamma is still kind of a proxy for the cellular response. It doesn't really look right at the cells, but still, that's in the right direction.
Culture the T cells and measure gamma as a
exactly.
often does. In fact,
Okay. Yes.
there's a paper out from Sean Crotty, and he sent it to me this week, which looks at T cell response. And I think we'll talk about that next week.
Yes.
There's also a new paper coming out from Rockefeller Group showing antibody responses. And, one of the things they say, these are really low, but I'm thinking, maybe it's the cellular response that's important. So we'll have that conversation next week. So now they have a clearance to begin a phase 2. So you have to say, okay, here's our phase 1. Can we do a phase 2 to the FDA? And the FDA said yes. And the clearance was made on May 6th. And so they're going to have 600 healthy people. And they're going to look at safety, reactogenicity, and immunogenicity of 2 vaccinations, 28 days apart, placebo 50 microgram or 250 microgram dose.
Yeah, so they're bumping up from the 25 microgram and adding and changing it to 50.
Yeah,
Right.
There's a thing in some stuff in here about how, based on these preliminary results, they want to include a 50. This is where it's ambiguous. They say they want to now modify the phase 1 trial to include a 50 microgram dose. That doesn't make any sense to me unless they're going to vaccinate some more people. But certainly they want to stick the 50 microgram dose into the phase 3 trial, which makes sense. So it says to me, or were they dropping the 25? I think so.
I think
they're just
so.
going to do 50 or 250.
Well, 50 and 50. Oh, you mean in the extended phase 1? Or in the phase 3, what I read was that they expect to use a dose between 25 and 100, but I think
actually,
they haven't gotten there yet.
Each placebo 50 or 250 in this paragraph. I don't know. Oh, yeah, but then it says 20. There's inconsistencies here. They say each subject will be assigned to receive a placebo 50 or 250. In the next section, it says the dose expected to range between 25 and 100 micrograms.
It's for phase 3. Phase 3.
For phase 3.
Right.
Well,
right.
Which I would not be surprised. So the phase 3.
Oh, for the phase 3. Okay.
Which is not starting yet. I would not be surprised if they change that based on what they see in a phase 2. And in a phase 2, they will immunize or not, and then put them out in the world and see periodically who gets infected and who doesn't. So that is at least 6 months, right?
Well, okay. So I looked back. I've been corresponding with a friend of mine and reading some of the lay press. Actually, this gentleman is a businessman, so he sends me articles from Market Watch and says, "What do you make of this?" And it was actually one pretty good article on the chimp thing that interviewed some of the people at Oxford in that study. And that study is, their phase 2 trial is designed for perhaps getting results in 6 months. But one of the things they say is, depending on the transmission, that is how much infection the participants might encounter, they might be able to see data in as early as 2 months. Okay. And if they go on that and they say, okay, that could be as early as 2 months, that might be justification for even getting the jump on a phase 3 trial. So maybe you could have results or early results from a phase 3 in as early as 4 months. And if that's the case, maybe you could be thinking about mass vaccinations by the end of the year. Now, I think that is borderline fantasy land, okay? But that's what they're saying. And I have to say that I'm really impressed with how fast all this is going. I never expected it to go this fast. But I also think that its caution is important, and we don't want to rush this. I wouldn't, and I don't know. I've been so wrong at so many phases during all this. I don't want to make any predictions. Some of the people involved in the process, and I don't think they're just hyping it, say, if everything went absolutely swimmingly and everything was working really great, we could be maybe looking at a fairly large scale beyond phase 3 vaccination by late this year or early the following year. But I think things would have to go at unprecedented speed and be really very positive in order for that to happen.
I wanted to make a couple other points. Why would you want to do 50 micrograms instead of 100? Two reasons. If there are side effects, you might get fewer of them with the lower dose. And also it increases your capacity to provide vaccines. So they're estimating that if they had a 50 microgram dose, they would be able to make 1 billion doses per year. And obviously then if it's 100 microgram dose, then that's only 500 million doses. And if you have to give it twice, that uses up more of the doses and so forth. And then in terms of side effects, what I read was one patient had redness, and then 3 at the 250 microgram dose, the highest dose, had some systemic symptoms after the second dose that were transient, and all of them were self-resolving. And there were no grade 4 or SAEs.
Serious adverse effects.
Yes, thank you. So that's promising. So this link that we have in the show notes is not the press release itself. I think it's an article by a company. So I didn't see the press release.
Same here. And you can see various versions of the press's spin on this. I don't know what they're reading, but I haven't seen the original press release. Actually, probably ought to look up on Moderna. It's probably there.
Probably.
I just wanted to point out that one other thing is that they talking about production. Yes. They're in a 10-year agreement with a company called Lonza, which is they're originally headquartered in Switzerland, but they got partnerships all over the place. The production they're imagining going on in the U.S. I think the plant's in New Jersey, Vincent. I have to look again. And so they are the ones that are ramping up production. And I did a quick calculation. If you had a 50 microgram dose that you had to do a dose and boost with, that's 100 micrograms per patient. 1 billion doses is 100 kilos of RNA.
So a lot of RNA.
And I still have the question that we discussed before is how are they going to make it? Is it chemical synthesis? Is it transcription? What? I don't know.
I'm worried about the scale up. I don't think it's trivial. Mm-hmm. And I think the lipids alone are not trivial. We're kind of not mentioning that, but these are proprietary formulations for these nanoparticles and not clear that they have all the raw materials they need. However, I hope they do. I'm not trying to be negative, but just there are a lot of things they don't tell us because they want to be secret and so forth. I do want to point out, though, that BARDA has invested $483 million in development and manufacturing of this particular vaccine. So not trivial.
Well, here you go. Here's the press release. And shame on us. We should have had this in here and read this, but I will put the link here and we can read it.
And have it for the listeners for sure.
Yeah, have it for the listeners.
Put it in the show notes. And if anybody finds something we missed, just let us know. We can come back to it.
And you mentioned Trina Sedaris, who sent this to you in the first place. And I just wanted to point out to listeners that she was on TWiV 149 in September 2011 at an ICAC meeting with Vincent Rich and Mark Palanch, poliovirologist from CDC, was another one of the guests, I guess.
That started a firestorm because she said, I check my stories before I release them with scientists and people. And then so many journalists said, I don't do that.
Yeah.
It was funny. Sorry, I interrupted you.
I do remember her saying that. I didn't remember the firestorm.
I remember that TWiV well. That was great. She was an awesome journalist. Still is, I'm sure.
Well, she's left journalism because it's not a very secure profession, I guess, but she's still in Chicago and writes periodically. Her son is addicted to TWiV, apparently, and she said he just sits there refreshing the page to see when a new one is coming out, which I'm sure is not true.
He should get the email links.
Should.
Yeah,
Trina, if you're not listening and if your son is listening, Trina's son, say hi to Trina for us. Tell her to listen.
Yeah, but listeners might want to know that you can sign up to get an email for when a new episode is released.
She sent me earlier in the week. So this is why science by press release, and that's her phrase, by the way. She said that is not good because earlier in the week, before the official, I'm not sure what the timing was, but the news all of a sudden was only 8 people responded to the RNA vaccine, which is not correct. Everyone responded, but I think the 8 meant probably neutralizing antibodies or something or levels. It wasn't clear until the actual press release came out a day later. So you have to be very careful unless you see the data. Even a press release is what the company wants you to see. It's not the paper. It could be different. We had an email from John about this. John is a technologist in Cambridge who does electronic stuff, but is an amateur molecular biologist. And this sentence I love. While some folk might watch The Sopranos, I might watch Mr. Racaniello's lecture series on viruses. Yes, that's really what it's all about, John. If you need to binge, most people will binge on The Sopranos, but if you binge on my lectures, I have incredible respect for that.
I just had a skim of this Moderna press release, and it looks to me like I don't see anything in it that we didn't cover.
So John had 2 questions. What's the mechanism of cell attachment and entry? Do the nanoparticles include attachment proteins embedded in the lipid membrane? And if there are attachment proteins, is there a risk of an adaptive immune response to them? And so, of course, the answer is there are no attachment proteins, because that would be an interesting, you would want to make an adaptive response against if it were the spike, but they're not included. It's just a lipid coating, and this attaches to cells by virtue of its charge and hydrophobicity, and then it's typically taken up by endocytosis. I found a lovely article, which, yes, has a lot of detail, but, I like detail like that. It's great about what it's made of and how they're different cationic lipids, phospholipids, cholesterol, lipid-anchored, polyethylene glycol. A lot of people have worked very hard on this for many years to optimize it. And they bind to cell surface. They're taken into cells by endocytosis. And then the RNA is released. The negatively charged phosphate group appears to be involved in cationic charge neutralization, and which is important for phase changes and endosomal escape. So somehow it escapes the endosome and ends up in the cytosol where it's translated. And then one of the interesting aspects of this is the article I found. They said the most important targets for these mRNA vaccines are the antigen-presenting cells, like dendritic cells, macrophages. In those, the mRNA is translated to protein. The protein is processed by the proteasome into peptides, which are then displayed on the cell surface in MHC1 molecules, and then they can be recognized by T cells and lead to generation of responses, antibody and cell responses.
So that article has a figure 1 that doesn't show the lipid nanoparticles in the same way as this new figure that's been pasted in.
I pasted that new figure in because of the article above it, because the article above it is a liposome, and it shows it fusing with the vesicle, and I don't think that's right based on my understanding. The nanoparticle is like I say, a half a leaflet surrounding a relatively disorganized lipid RNA complex in the middle. Yeah,
this picture is really cool. So if you can provide the link for that, we should have that in the show notes. The liposome versus lipid nanoparticle.
It's the Journal of Google.
Okay.
It's a thing.
Here,
yeah, okay. We'll get it.
Cool. I mean, we can even use Google, okay? You just, because, here we go.
Well, Google will always give you a journal article. It's good. That's okay.
Yeah, that's really a good, picture of what the lipid nanoparticle is. Very different from a liposome. All right.
And so, Yeah, Vincent, you're touching on the stuff I wanted to talk to Brianne about. Having the
okay.
protein made in the person being vaccinated, if I understand it correctly, helps stimulate the cellular as well as the antibody response. Okay, which is a good thing. Whereas just delivering the protein, if I understand it correctly, has a tendency to skew things towards just the antibody response. Am I right?
It's kind of the difference between the Salk and the Sabin vaccines,
right?
In terms of the response that you get, if it can make something within the cell. That
having been said, we don't. There's two issues. One, again, we don't know what the correlations of protection are. Correlates of protection are. So you don't really know if you need a cellular response. I can't imagine it wouldn't be beneficial, but you don't know. And the other issue is that in terms of these side effects that we've been talking about, the disease enhancement, as I understand it, the balance between the humoral and the cellular response is going to be important in determining whether or not, if it's going to exist at all, that sort of side effect is going to happen. So these issues become really important.
All right. One more brief note. I just stuck this in this morning, so I'm sorry about that, but I woke up in the middle of the night thinking this would be interesting to mention. It's a journal preproof. Okay, what is a preproof? Not a preprint. A preprint hasn't been reviewed yet. A preprint has been reviewed, and it says it has been accepted, and this is the journal Developmental Cell, but it's not yet been formatted. A preproof.
So it has been peer-reviewed and accepted, right?
That's right. Nels and I yesterday on TWiV we did another pre-proof from another journal. It wasn't clear at what stage it was, but here they say it's been accepted, but it's not been put into the nice formatting. Okay, so this is a developmental cell. Cigarette smoke exposure and inflammatory signaling increase the expression of the SARS-CoV-2 receptor ACE2 in the respiratory tract. And kudos to the authors for not saying the COVID-19 receptor. Kudos. And a really, actually, very nicely done paper, very carefully presented, nice data, comprehensive, and they recognize its limitations up to a certain point, which I will, of course, tell you in a moment. But this basically shows that in a subset of secretory cells in the respiratory tract, they look at rodents, they look at human lungs, that chronic smoke exposure actually increases the expansion of this cell population and increase in ACE2 surface production. And they look at, mouse models. They look at data that is there from smokers and non-smokers. And, if you quit smoking, the ACE2 goes back down again. And in fact, ACE2 goes up not just in response to smoking, but some inflammatory stimulus. So if you get an infection, it seems to go up. And it's been suggested that actually ACE2 is an interferon stimulated gene, an ISG. So one of the reasons I really liked this paper is because most of the work is done looking at levels of RNA, right? It's very easy to do that. ACE2 RNA. And they say, it may be that doesn't result in increased protein. Hallelujah.
Yeah.
Yes.
Good for them.
Good for them. I don't know if they were made to put that in or not, but they try and address it. They do an experiment to say, yes, the RNA and the protein levels correlate. It's a very important point that a lot of people don't even address. So that's one of the reasons I like this. So I guess the idea is that smoking could increase your susceptibility. And there are some epidemiological data, suggesting that smokers have more serious disease. So could this explain it? I don't know, because how much of a difference is receptor level going to make, right? If you have, that's the question that needs to be answered, which they don't address at all. Does increasing the receptor level make a difference? And someone needs to do the experiment where they say, okay, if we have one 10 receptors on a cell versus 100, 1,000, or 10,000? Does it make a difference in the cell's ability to get infected or something else? I don't know how else but that's the important question. I mean, this doesn't answer it. All this says is smokers have higher ACE2, but whether that matters is, I mean, I wouldn't assume that just having more receptors is going to make a difference, because it may already be at a level that is enough to let the virus in. It's so anyway, I like the paper, but I think that's the main question. We shouldn't conclude from it that smoking is increasing your susceptibility via this mechanism.
I didn't hear. Did you say anything about the authors or where they're from?
I didn't, no.
Okay, so Joan Smith is the first author. Jason Scheltzer is the last author. And the two of them are at Cold Spring Harbor, although Joan Smith is maybe now or also both at Google Incorporated in New York City. And then people from Johns Hopkins and Hofstra as well. It's in developmental cell.
But Kathy and Rich, the idea that more receptors would make a difference, that's not clear, right?
No, I don't think so.
Right.
I mean,
I agree.
You'd have to do the experiment.
Yeah.
And I don't know how you do the experiment, okay? Because I'm not going to trust an experiment in cell culture.
Yes.
You'd have to, I suppose, use an animal model. Mice lie.
Yeah.
Really hard.
Monkeys exaggerate. 4
different transgenics with different levels of ACE2.
Yeah.
Really hard,
I don't know.
Right? So anyway, it's interesting, but just really, I would say don't smoke.
Yeah.
That's a good idea.
It's probably good not to. Oh, this is an interesting picture.
Yeah. Well, they talked about how they did these experiments in mice, and I immediately imagined mice smoking cigarettes. And of course, if you Google mouse smoking cigarettes, you can find images of mice smoking cigarettes. So there's one.
There you go. Okay. I'm sure we'll hear more about this. All right. So let's do some email. The first one is about our, TWiV this week. I finally got around to releasing some TWiVs recorded last year. I'm going to try and release one a week. Otherwise, they're never going to get out. And the one I released this week.
Well, and many of them turn out to be presciently appropriate.
Mm-hmm.
Yeah, interestingly, what's interesting is that, so this week's was an interview I did with Peter Daszak of EcoHealth Alliance, the nonprofit that has been involved with wildlife sampling, in particular, many parts of the world, but in China, and they were helping get those viruses from bats that are highly related to SARS-CoV-2. And he wrote to me just this morning. He said, it's pretty spooky to think while we were sitting there, the virus was already circulating 1,000 miles away.
Yeah.
We had no idea. This was early December.
Yeah.
Anyway, he's coming back next week. Is it next week? Let's see. No, the week after. June 2nd. And we can talk to him more specifically about SARS and bats. Anyway, Maureen said, I freaked when I saw today's episode. I love these guys. And that's EcoHealth Alliance. And she said she sends a link to a YouTube video of them from 2004 showing how they go into the field and get it. Stay healthy, keep teaching.
It's an awesome video from 60 Minutes with Peter Daszak and several others. And also, I can't figure out how they got such good audio. They have audio when people are wearing PAPRs and are in BSL-3 suits. They're on a boat that's, going down a river or something. They're hiking in a forest, and the audio sounds like they're in a high-quality studio. It's amazing.
So they put lavalier mics on them, right, which are wireless.
Yeah.
So they can be under this. In fact, I think in the needle video, they put some of those on us under our suit so that they can hear it. And it's very good because it's clipped right onto your shirt. Right?
Mm-hmm.
And you can do it on a boat, in a forest. And, all these locations are pretty quiet. So it works.
But the boat is not quiet. I mean, so it just, anyway, I was impressed with that. But it's very cool to watch.
So that's where you have professional audio people helping out, as opposed to me, who tries to do everything himself. And sometimes it works and sometimes it doesn't. But that's what we're stuck with for now.
Right.
Okay, Kathy, can you take the next one, please?
Sure. Izzy writes, hello, Twivers. This is a critique of some of the data coming out of the Jenner vaccine trial in monkeys. And that was as we talked about last week, the chimpanzee adeno vaccine. It was discussed on TWiV 613, Jenner vaccines and COVID-19. It's a very rhymey thing. And Izzy is referring to the Hazeltine article in Forbes, which is essentially I think kind of an opinion article. The issue, as I understand it, is that the vaccinated monkeys showed differences in the amount of viral RNA on bronchoalveolar lavage, but not on nasal swabs, and the titer of neutralizing antibodies was not high. Taken together, this data seems to suggest we can do better. So I took a look at the article too, and he says it is crystal clear that the vaccine did not provide sterilizing immunity to the virus challenge, the gold standard for any vaccine. It may provide partial protection. So it reminds me of something that somebody said a couple weeks ago, that, the best vaccine, the first vaccine out may not be the best vaccine. And if one comes out and you can easily make 1 million doses, and then 3 months later, another one comes out and you can make 1 billion doses, it's all good, but we just have to have many different things, darts that we're throwing at this dartboard, if you will. And so when this was mentioned sterilizing immunity, it reminded me of what I had just listened to yesterday, which is the Immune Podcast 31, where at 54 minutes in, a listener asked, do vaccines work at least through the mechanism of increasing herd immunity if they don't elicit sterilizing immunity? And so the discussion concluded that you probably can reach herd immunity even if there isn't sterilizing immunity. So Hazeltine's thing of it's not the gold standard, it may provide partial protection, and that may be good enough in the absence of anything that's better. And Vincent pointed out that you don't get sick, but you get infected with repeat doses of some of the common cold coronaviruses. And so sterilizing immunity is not getting infected, no replication, no symptoms. But non-sterilizing, there could be low-level infection, mild symptoms, and potentially the virus is not disseminating. And if people do have some of this non-sterilizing memory response type of immunity, you can reduce the viral load. And this is where Cindy said, yeah, it's like if a non-immunized person, say, is shedding 100 viruses, a person with non-sterilizing immunity might only be shedding 1 virus. And that made it really clear to me, because I've never really had a good grasp on sterilizing versus non-sterilizing.
I think this is a bit of a negative article, which is not fair. The name of it, did the Oxford vaccine work in monkeys? Not really. That's just a misrepresentation of the data. Okay. And I will not tell you stories about Bill Hazeltine, which will cloud this. But first of all, sterilizing immunity as a gold standard. Folks, IPV, inactivated polio vaccine, is not sterilizing. The virus replicates very nicely in your gut. It doesn't go very far once it gets into your blood, but it's not sterilizing. And we're still using it today to eradicate polio. And as Kathy said, the common cold coronas don't induce sterilizing immunity. So maybe a vaccine against SARS-CoV-2 will not. But more importantly, the monkey data is very interesting, that the monkeys, the lower tract is protected from virus replication and disease. But the upper tract is not in the vaccinated and control groups. You get a lot of upper tract replication. And there is an article in which one of the authors, Vincent Munster, and in fact, the senior author on that paper, Emmy DeWitt from Rocky Mountain Lab, is going to be on in a couple of weeks, so we can talk to her about this. But they said they challenged him with a huge dose. You don't have the luxury of having hundreds of non-human primates to give in different challenge doses, right? Mm-hmm. Because they're expensive. You have to pick your dose. So they said, let's go in with a high dose, because if it protects at a high dose, it'll be informative. I can totally understand that logic, if in fact that's what they did, but that's the way I'm interpreting it. And so in that case, they get high upper tract replication. I think they put the dose into the trachea, right? So, and Munster explained in an article that it probably isn't a relevant situation because in humans, you get an upper tract infection with a lower dose, and so it might be very different. But I think if you look at the data, it did protect them. It induced antibodies. The antibody levels are low, yeah, but that's what we're seeing in recovered patients too. So I don't think it's all as negative as that Forbes article would suggest. I don't know why he's looking at it that way, but I don't think it's the right view. Rich, you were going to say something. I'm sorry.
So sounds like I need to listen to Immune 31.
Mm-hmm.
There's been a lot of, I've been wondering about this sort of the definition of herd immunity because it seems like in many circumstances, people equate herd immunity with exposed individuals experiencing sterilizing immunity. And they say that since this vaccine or since this virus may not give sterilizing immunity, we can't talk about herd immunity. And it sounds like that's not true. That depend if the nuanced definition of herd immunity can include immunity that's not sterilizing. Am I reading this correctly?
I would think so. IPV gives herd immunity without sterilizing immunity, right?
Okay.
I think the key is if it prevents disease.
Right.
Not. And so I don't think you should you should expect a vaccine that's going to completely prevent replication. I think a lot of them don't do that. And so I would look at sterilizing immunity in terms of, I would just look at immunity in terms of prevention of disease.
Right.
And
I would even go so far as to say prevention of serious or fatal disease. I don't care if I get a cold.
And that is subject to herd immunity concepts for sure. Right?
Okay. All right. Good.
Rich, you're next.
Ken writes, "Hi, crew. I've been listening to VR's lectures and TWiV since early 2018 by a chance discovery when I found that his lecture series were understandable even to numpties."
Mm-hmm.
What are numpties?
Well, I had to look it up, and it's pasted down below. It's informal, British, a stupid or ineffectual person, origin 1980s, of Scottish origin, perhaps an alteration of numbskull, with the ending remodeled on the pattern of Humpty Dumpty. So, Vincent's lecture series is understandable even to numpties.
Ken, go easy on yourself, okay? It's okay, right, dude? I think if you're curious, you're not a numpty.
Right.
Thanks for your considerable labors, and please keep it up. I recently had someone forward an item by Professor John Ioannidis. I think it's Greek. It starts with an I, capital I. Oh, yeah. Pronounce that again, Kathy.
I think it's Ioannidis.
Okay, Ioannidis.
Something like that.
Who espouses that the whole reaction to SARS-2 has been overblown and out of proportion to its real threat when compared to similar viruses. He establishes this by application of Bayes' theorem of probability in many of his other works. He has a brief outline for the hypothesis. It's dated April 3rd, 2020, and considerable data has come to light since then that could be inserted into Bayes' formula, the basis for this opinion and conclusion. Can this be used as a valid academic source for conspiracy theorists? Your comments, please. Yeah, I have a really strong comment, and that is that if you want to say that this has been overblown, go spend some time in the hospital with Daniel Griffin. Yeah. Okay? People are dying from this, and they're dying from it in numbers that are overwhelming the healthcare system where appropriate precautions haven't been taken. And you can apply all the math you want to this, and it doesn't touch on the experience of the people working in healthcare or the people who are dying on it. Yeah, you can put a bunch of stuff on this and say, well, if you extrapolate on the infection fatality rate, it's only like 0.4%, and that's, not many. That's hardly comparable even to flu in a bad season. Nonsense. Okay. That's what I was saying months ago, but nonsense. If you look in the hospitals where people are really under a lot of stress, this is a serious problem. And if you extrapolate that case fatality rate to a situation where, in the absence of a vaccine, we have to establish some form of herd immunity, you're looking at on the order of 1 to 3 million deaths in the U.S. Okay, deal with it, dude. I don't care about your formulas.
So yes, he is probably he's a dangerous academic source for the conspiracy theorists. He is a physician scientist in California, maybe
Stanford.
Stanford. Yeah.
I agree with Rich, and I just want to add that someone else weeks ago wrote a letter saying we shouldn't be so hard on Ioannidis because we had mentioned this before, and but he has published papers saying most science can't be reproduced, and I think this is just nonsense. And I don't care what his credentials are; he's way off base both in that and this as well. This is wrong, and so I don't think we need to consider it anymore. But Ken, I will point out from Brisbane, where on the 19th of May, they just attained 100 deceased from SARS-CoV-2. 100. Yeah. That's pretty low.
Only 100.
Amazing. It's deceptive because there's significant regional variation in this. And at this point in time, people's experience in different areas is quite different. Granted, I've been locked down, so I really don't know what's going on. But my sense is that in Austin, for example, it has not yet been a big deal. So it's easy to get complacent, and it's easy to say, this has all been overblown. But it's clear to me, looking at the places where this has really hit hard, that this is a serious, serious problem. And when you extrapolate the numbers that we do have, this may be only the beginning, dude. Mm-hmm. What's the percentage of people who have seroconverted so far? We don't know, but I'd be surprised if it were as much as even 20% on average throughout the U.S. And if we have to establish some sort of herd immunity for this simmers down, then we have a long way to go.
All right. Mason writes, hello from sunny Florida, 73F. Our university is investigating how social distancing might be possible in the classroom by putting small classes into big rooms to enable 6-foot distancing. When asked if they had considered airflow, the person in charge of health and safety expressed skepticism that airflow in classrooms had significant effects on the spread of viruses. Are they right? Skepticism. Are there any studies that can either reassure students and faculty that this isn't a factor to be concerned about, or, as I suspect, studies that could help inform our administrators to take airflow into account when deciding how many students a classroom can relatively safely accommodate? All right, so Kathy has a lot of comments here, but before she does that, let me just say that I think last week we had an article sent in from a Chinese restaurant where, all the infections occurred at a table beneath the air conditioning vent, right? Something like that. Do you guys remember that?
No, I don't remember that. But
yeah, I do remember, and sort of the picture of it, and people on either side.
Yeah.
Family and so forth. Yeah.
So there are things like that, but I think in the absence of evidence, you should assume it's going to happen and take care of it. What do you know, Kathy?
So I wrote to a couple colleagues, and in some places, at least, industrial engineers are being consulted. And I think we even had email from an industrial engineer about airflow and things maybe after the Chinese restaurant discussion. But anyway, one statistic I've heard is that, for instance, there would be 75 people in a lecture hall that seats 400. And overall, this probably relates to the droplet versus aerosol question, which I think is maybe still not resolved. And I told you that Christiana had said that Christian Drosten mentioned on his podcast that he thinks 45% of transmission is aerosol, 45% droplet, and 10% fomites. And so I checked with her again, and she sent me the excerpt from the transcript in German, which I haven't looked at, but she summarizes it where he says it's this back of the envelope calculation, taking all the studies together that give that kind of data. Or that he says half is aerosols, half is droplets, and 10% are via contact. And so she calculated that he meant 45% because it couldn't add up to 110%. And then she looked at some modeling articles from Christoph Fraser, and she gave us the link for that. There's at least some support for the roughly 10% environmental transmission. So that may relate also to this lecture hall question.
I think that the person in charge should not express skepticism without the data, but I do think that couldn't you imagine filtration in the airflow systems that would reduce the droplets, for example? It seems to me that wouldn't be a difficult thing to do.
Yeah, in fact, I go back to the podcast with Michael Schmidt, where he talked a lot about aerosol transmission, and he talked about engineering standards and numbers of air changes in various rooms for various purposes, and whether or not HEPA filters were involved. So there are measures that can be taken to try and mitigate problems where one anticipates that there could be aerosol transmission. And I think that that's probably a reasonable consideration for, you ought to get if you're talking about reconvening your classes, I'm sure that there are public health officials with engineering backgrounds who can come in and talk to you about the impact of the air handling on the possibility of transmission of infectious disease. I'm sure think
that you should have all the students wear face masks, no matter what you do. If you're not thinking of that, you're way off.
Right.
And if it were up to me, so you have a lecture hall that seats 400, you're going to put 50 or 75 students in. You have to, I would say, rotate the class in and out and have the others look online. So one day you're online, next day you're in class, and so forth. That's one way to do it. With face masks, I think you have to think of these things, but nobody asks me. But I'm just giving you my opinion here. Kathy, you're next.
Okay. So the next three are about choirs. Two are about software, and one is on a fluid mechanics study. So the first one was from Jameson. Alan Dove read my mind when everyone was discussing audio communication services. As a PC gamer, I was about to write an email when I heard the program Mumble mentioned. And then Alan buzzed in with the correct answer that it's also indeed the best audio communication service, gaming or otherwise, with the lowest latency. So you can hear everyone in real time. And then he comments on the fact that he's been listening that since 2012, when the TWiV crew reviewed the movie Contagion, and he watched it just so he could listen to the podcast. And he said, if you don't recall, you were all very hard on it back then, but I just rewatched and was pleasantly surprised how much, in the current context, it got right for a mainstream movie. I'd say it's definitely overdue for a revisit. Also, I'm glad to see this podcast and Labor of Love getting a fraction of the attention it has always deserved. I've shared it with my whole family during the quarantine, and my parents are especially appreciative. This continues to be such an invaluable, transparent discussion of information between so many well-educated people. Best regards, Jameson.
It's funny. Chorus is a bunch of people out there interested in that.
Yeah. Right? It's great. Yeah. It's brought all the singers out of the woodwork.
Right.
This is great.
Contagion. All right. Go back to
just go back to that TWiV episode. Yeah.
One vaccine tested in one monkey, and then the lady stuck herself in the thigh with the vaccine, and then the same day went to visit her father, and she was protected. Come on.
Well, it's unfortunate because I think that's where some of this idea, that kind of thing is what gives the public the idea that we can have a vaccine tomorrow. Mm-hmm. That it's all the process is all magical.
On the other hand, all these vaccine studies are all being criticized, right? Mm-hmm. Oh, not enough animals, not enough this. Are you kidding? Contagion only had one. That's all you need, apparently. By the way, I asked Ian Lipkin about that. Why did she stick herself in the thigh with a vaccine that was eventually intranasally sprayed? Okay. And he said, well, Vinny, here's what happened. I'm a script consultant, and I review it, and they have her sticking it in her nose, right? And then the actors get on the set, and they look at the script for the first time and say, I don't like this. I want it to be more dramatic. I want to stick it in my thigh. And I'm not. And Ian says, I'm not there. And the director doesn't know better, so he says, sure, go ahead, stick it in your thigh. And that's how it happens.
Right.
I believe him. Rich, you're next.
Oh, and it looks like that might have been TWiV 167. It starts with a cough.
Wow. That was a long time ago. I've never seen the movie, I don't think. Aaron writes, potential solution to chorus practices free. Audio latency is the bread and butter of my existence. I'm just, he says in asterisk, so he's been listening. I'm just an audio engineer and designer of audio recording hardware and software with a lot of attention paid to the big L word. I assume that's latency. Mm-hmm. When you are listening to yourself in real time, by around 13 milliseconds, most people notice a small delay. Oh, this is great. I'm getting all excited. Some are much more sensitive, and nearly all people are disturbed by it by 20 milliseconds. It's interesting that we are that sensitive. Even a single sample of latency, 1/44th, 100th of a second at 44.1 kilohertz sample rate, can cause some problems through phase cancellation if you can hear both acoustic and monitored source at once. What I get out of that is he knows what he's talking about, or sounds like it. However, after all that, when playing with others, assuming the instruments aren't played too quickly, some people can put up with 50 milliseconds or more of latency, providing they are not hearing their own monitored signal, but only the signals of those they are playing along with. As a however to that however, getting a constant 50 milliseconds across variable and questionable networks is a tricky thing. Ninjam was created over a decade ago to work out this sort of problem for free. It integrates easily into Reaper as well in the event you want to record it all, or if you would like to use real-time audio processing alongside your virtual choir practice, and gives a link to Ninjam. Ninjam operates in fake time so that all participants are synced to start at exactly the same time, but won't hear everyone until the second go-round. The Ninjam client and streams
records and
streams. This is. Did you paste this in, Kathy, or is
no?
This
I think it's part of his letter.
Okay, yeah. And this is a quote. The Ninjam client records and streams synchronized intervals of music between participants just as the interval finishes recording. It begins playing on everyone else's client. So when you play through an interval, you're playing along with the previous interval of somebody else. And then you're playing along with your previous interval. If this sounds pretty bizarre, it sort of is until you get used to it. Then it becomes pretty natural. In many ways, it can be more forgiving than a normal jam because mistakes propagate differently. I will have to look into this. This is all very interesting. Mm-hmm. This is all. We were just talking. Our chorus met virtually last night, and there are a number of individuals looking into software, hardware, etc., to see if they can help us out. So I will. Matter of fact, right now I'm going to copy this letter into a blank email and send it to him. Mm-hmm. Thank you, Aaron.
This Week in Chorus.
Yeah.
Aaron, you'll be interested to know that We typically record TWiV and all the other podcasts. We have people on individual Skype connections that I then mix together, but I provide each person with a mix minus that subtracts their own feed so they don't hear themselves with latency. In other words, you'll speak, the sound will go through the system and back to them. It will be over 20 milliseconds. It would be an echo, which would be very disturbing. So I pull that out by subtracting everyone's microphone from their own feed. And so they're only hearing other people talk. They hear themselves in their own headphones because that is local, but their signal is not being routed back to them otherwise. So that's a mix minus.
Wow, I didn't know you did that, Vincent. Thank you.
I didn't either. So, but you said Skype, but now we're using Zencastr. Is it the same? You do this mix minus?
So Zencastr is doing it automatically.
I see.
For us. So I don't have to do that, because the way I did it in my office was to have a physical routing of cables. I have a big mixer in my office, and I have to have a special cables to pull yours out and so forth. It's a bit complicated, but Zencastr now does it automatically, and that's the way to do it. Yeah. But you'll notice that sometimes when we're starting, we have an echo, which is basically latency, and that there's a setting for removing that specifically in Zencastr.
Oh, I remember when that was happening, and then you just did something and it went away. Yeah. Yes, perfect.
They're mix minus equivalent. Yeah. And then we have another one from Janet, who sends another choir story from The Guardian. It looks like you have summarized this, Kathy, right?
Indeed. So this is talking about the Amsterdam St. John Passion concert, Bach concert, March 8th. And so the details of that, and at least two other choral things happening, one in Yorkshire, and I forget where the third one was, are mentioned in that whole article. But the key thing here for me was that a fluid mechanics expert is cited in this article in The Guardian, Professor Christian Kehler of the Military University Munich. And he conducted experiments because he was doubtful about the dangers posed by concerts. He studied singing in low and high frequencies and all sorts of things like that. He also studied different instruments. And based on the flow analysis they did, he found that one or two instruments did pose threats in terms of their powerful airflow. Flute, especially, also oboe and clarinet. However, he found that singing is quite safe. Air was only propelled about half a meter in front of a singer, and that is not far enough to cause the infection levels of these outbreaks. He thinks that the virus was probably spread among chorus members because of their close proximity. These outbreaks all occurred during the early days of the COVID-19 pandemic, before lockdowns were imposed, before our minds were concentrated on the importance of social distancing. And I would add, and before anybody was thinking about wearing masks to protect everybody else. He goes on, choir members probably greeted each other with hugs, shared drinks during breaks, and talked closely with each other. The evidence for a link with singing and spreading the virus may look compelling, but it is still anecdotal. Without data from comparably large groups who interacted in the same way but didn't sing, it's hard to be certain that the singing was responsible. And I would just give a caveat to this. This is a Guardian article citing this fluid mechanics expert, and whether that fluid mechanics expert has published anything and whether it's peer reviewed, I don't know. So
didn't we have a study where the force of singing is propelling droplets farther? Do I remember that?
There was something about loud talking and
okay.
Yeah. So
I think that plus that we had the that I attended that webinar with the Gesundheit guy on it. I forget his name, unfortunately, but I think that there are reasonable data to suggest that singing and different sorts of talking and stuff can influence the aerosols you produce and how far that they go, and that, relative to just hanging out and talking with your friends, singing is, at least in theory, a riskier activity. So, and a lot of what he's saying here is not, it makes some kind of at least logical sense. And in fact, the CDC article that the MMWR, Morbidity and Mortality Weekly, that article that describes that choir does point out that the individuals involved were not taking any precautions, and there was plenty of opportunity for fomite transmission and direct contact transmission, and et cetera. So I think it's not incorrect to say that we don't have data from this sort of gathering done with whatever social distancing precautions you can implement under those circumstances. But I would say I'm not willing to do the experiment.
Right. You might also think about, though, if you had 75 people in a lecture hall that would seat 400, and if people were wearing face masks and didn't hug and didn't share their pencils and all those things that you might do in a chorus rehearsal. And since we brought up the MMWR thing, I just want to make a point again, because Vincent used that CDC image from that paper as a show image for that episode, that TWiV episode. And it shows a stylized poster of what the choir could have looked at, with the one infected person in the back right corner, house right, not house right, stage right, and then everybody else in between being infected, and then a few people in the front row not being infected. It's all just done to show it to you graphically, and it's not the seating chart for the actual chorus, because they did not publish that in the paper intentionally, because you can imagine that at least the one person who was sick knows who they were, and probably most other people in the choir would be able to figure it out from a seating chart. So,
yeah. By the way, is stage left different from house left?
Yes.
Yes, they're opposite.
So house is when you're sitting in the audience.
Correct.
Which makes sense to me. This actually makes sense. And stage is when you're on the stage, right?
Mm-hmm.
Wow. Something makes sense. Some vernacular thing makes
the other stage direction you got to assimilate is upstage and downstage.
Mm-hmm.
Downstage is towards the front. Upstage is towards the back. So when you upstage somebody, okay, you stand in between them and the audience, which makes them upstage relative to you.
Okay. Makes sense. And then you say, don't effing upstage me, buddy.
Exactly.
Okay. That's where the vernacular comes from, right? Mm-hmm. Upstaging someone.
Mm-hmm.
So there we have a huge segue into singing. That's not completely irrelevant actually quite relevant to the whole coronavirus thing.
Right.
It's quite interesting. Yeah. All right. I'll take the next one. Okay, Kathy,
sure.
Since you did that. It's from Eric, again, from South Korea, South Korea, of course, wonderful job containing the outbreak.
And Eric wrote us before with a detailed explanation of what his experience was like and how they did it.
New cases have been generally falling down to single digits for a couple of weeks before there was a cluster with over 100 recently centered in Seoul clubs and bars. They rapidly tested 45,000 potential contacts, and new cases are again trending downwards. Trace, test, trace, identify. Is that it?
Isolate. IS.
Test. Tetris.
Mm-hmm.
It's going to become a thing, Kathy. Tetris.
Yeah,
you'll see it.
Absolutely.
Alan tweeted it. So yeah, it's already, and then somebody picked it up and put it on Facebook. So yeah.
Yep.
It's going around.
And now pretty soon it'll be on CNN. They'll be using it. And it started
and Alan credited at Virus Prof Sings. So CNN better do that too.
Someone wrote that every time they see something on news, it's already been on TWiV, which is okay. That, we get things first.
Yeah.
I mean, Daniel Griffin appeared on TWiV, and the next day he was on CNN.
Mm-hmm.
There you go.
I do think there's a link. Let's see. They rapidly, 45,000. As a result, tomorrow schools will begin to reopen, which leads me to my question. As a high school teacher, I have been following the announced measures being taken at schools throughout Korea, including opening in phases, 1 grade a week, requiring all students and teachers to wear masks, restricting use of air conditioning and other air circulating machines while keeping windows open for ventilation, increasing time between classes so students can clean their spaces with alcohol wipes, use the restrooms in small numbers. Lines are taped on the floor outside restrooms to maintain social distancing for waiting students. Eating in 15-minute shifts in the cafeteria with all surfaces clean between shifts. Students sit in a zigzag pattern with alternate seats empty, and plastic dividers have been installed to create individual eating areas.
Ooh.
Taking the temperature of each student at least 5 times a day.
Whoa.
I've also been completing self-checks for the past 2 weeks in preparation for returning. Immediately moving any student with any respiratory symptoms to a special isolation room for evaluation. Requiring students who are or whose family members are possible contacts to stay home for 14 days. Placing antibacterial film on high contact surfaces like light switches and elevator buttons. I think many of those are good. And I wish we were doing some of these. I'm not sure about 5 times a day temperature check.
But, a temperature is non-invasive. I mean, it's slightly invasive, but it's not bad, So.
The problem is, of course, that some infections are not having temperature rise. And so I guess you're going to just miss them.
Right.
And what's with the antibacterial film?
I never heard of it. Sounds pretty interesting.
But if it's only antibacterial and not antiviral, what good is it?
Yeah.
Well, I mean, Michael Schmidt would tell you that bacteria are a lot tougher than viruses, that viruses are wimps. And if you can kill bacteria, you can, quote, kill, unquote, viruses. They're already undead, right? But you get the idea.
There are other measures. Our Provincial Office of Education sent teachers a 200-page manual on such procedures. Oh, it's too long. Nobody's going to read it all.
Yeah, right. You need a cartoon.
I wonder if this may be a preview of what American schools will be like in the fall.
Not a chance.
Most of my students are worried these might not still be enough to protect in tightly packed schools with over 1,000 students and staff. Are they enough? What should we consider? And perhaps more a question for educators and parents than virologists. Is this any more sustainable than online lessons?
Yeah, good question.
So my view is that these are great questions. I think what you're doing there is great. I think we're not going to come anywhere close here. Some places in the U.S. might, other places won't because they don't believe that it's important to do it. This is what we're seeing here in the U.S. And I think you can do that for a while, yes, because you have to get the students interested in the material so it distracts them from all this stuff, and that's what you're supposed to do anyway. So I think it's better than online lessons, but that's my opinion. What do you guys think?
Well, I think I agree. I think this is tough. I think we got to go back to school, because I don't think we can, frankly, I don't think we can outrun this thing. Absent a vaccine, and there's no promise of that, especially by the fall, I think we're just going to have to suck it up. And I realize that this doesn't sound good to a lot of people, but in my opinion, at this point, the best we can do is do whatever we can to mitigate and try and, in particular, protect the most susceptible people and try and keep it manageable with respect to what the healthcare system can manage within certain practical limitations. And I think one of those practical limitations is somehow I think we got to go back to school. I just can't see staying locked down and forever. And at the same time, despite all these mitigation efforts, I don't think that the virus is not going to go away and the spread is not going to be zero. And when you get people back together in schools, there's going to be some spread in the schools. They minimize it. And when people are obviously have a problem, take them out and quarantine them and test them and do contact tracing to try and minimize things and protect their grandparents, like me, But I think you got to go back to school somehow.
Agree.
Yeah, I think it kind of relates to the question about lecture hall size and things like that too. I think that cautiously, people could go back to school with some of these kinds of things, if not all of them, in place, but with the understanding that things could get really bad and we might have to all go back to staying at home and distance learning again. And so I think it's hard work to prepare for both of those, but being a little bit glass half empty at this point, it seems wise to prepare for, maybe even just fall semester if that had to happen in K through 12 and universities. I mean, we might see a spike starting to happen in August, in which case, some places schools are already going back in August. I think test, trace, isolate, and be prepared to maybe have to go back to this again.
I think, Tetris plus hand washing, masks. It will be. Let's put it this way: it will be interesting to see how much mitigation we get out of those measures. I wouldn't be surprised if it would be pretty substantial.
Yeah.
In Paris, they sent kids back to school already, and they've had 70 infections among the kids, I think. They're reevaluating what to do. But maybe they didn't use all of these approaches, right? It's hard to do all this, but I think you should do it. I agree. Tetris and these things can get you back to school. And I agree, we have to get back because you can't, otherwise we're going to wait till next summer for a vaccine, and can't do that. Yeah.
I just wanted to comment on the temperature taking 5 times a day. It reminded me of someone I know is having some rescheduled surgery and said that they had to get a COVID-19 test, and it was the nasal swab, and said it was unpleasant, but not that bad. Men are just whiners. And just said, it's like, have you ever had pop carbonation go up your nose when you take a drink? So,
okay. So, I've been wondering about this because we've done a couple of papers in the now distant past, probably 3 weeks ago, that, if I'm not mistaken, drew the conclusion that saliva was every bit as good as a nasal swab. And yet, I haven't seen that take hold. This is maybe a question for Daniel.
Yeah, I think it is, but I agree with you. I mean, that one paper we did was a preprint out of Yale on saliva. It hasn't been published yet, as far as I know. And I mean, it really needs a bigger study because it was not that many patients, 100 and something. And so I probably this is going on, but we should ask Daniel. He would know. Yeah.
Well, and Daniel also even pointed out that if you just did a swab, not all the way back into the brain, as it were, but that also, gave decent results. Is there anything about having to adapt a test between a nasopharyngeal swab and a saliva sample in terms of the media or anything? I wouldn't think so. So that wouldn't be causing the delay. It's maybe what you said, that it just hasn't been peer reviewed, and
Dan always says that swabbing is always inconsistent because different people work differently, and so that's why the repeated swabs are hard to get a reproducible finding because different part of the not your entire mucosa is not infected; they're just patches most likely, and depending on where you go. But saliva is great because it's a fluid. It's like blood. You're going to get a sampling of whatever is in the mouth. And it just seems to be theoretically better, right? And easier. Not just easier, but better in terms of reproducibility. And that's what that study said. So I don't know why we're not switching over to it. And, Daniel had also talked about a nares swab, which his organization did, and said it was just as good as a nasopharyngeal swab. You just rub the nares with a swab. So we'll ask him this week, I guess. All right, let me just take one more, and then we'll stop because I have a meeting at 1, unfortunately. And this is from Sharon, who's an infectious disease fellow at George Washington University, and wants to know about using Maraviroc for treatment of severe COVID-19. And Maraviroc is a drug, an HIV antiviral that blocks the co-receptor CCR5, so it's an entry blocker. And I have no recollection of this being used for COVID-19, and I can't imagine that it's blocking virus entry because the CCR5 is not involved in SARS-CoV-2 entry. But as Sharon said, how might it modulate the immune response? So CCR5 is a chemokine receptor. And so I am not aware of how that would impact COVID-19. And I looked and I couldn't find anything. I know we mentioned it once because I remember Rich was reading the letter and he couldn't pronounce it, and I had to help.
I remember that too. Yeah.
But I don't, and I guess I could search all the letters, but I haven't done that. But I'll ask Daniel this week as well. All right. So let's stop there because I have an IBC meeting.
Fun.
And I really don't want to go, but I really should because there aren't many. I'm the only virologist on the. Well, actually, there are 2 virologists, but I feel I should go. And then I have a faculty meeting at 2, so I want to take a little break here, a bio break, as we say at meetings, at study sections. Can we have a bio break?
Yeah. And maybe eat a little snack or lunch or something.
I should eat something. Yes. But when I eat, then I can't speak properly for a few minutes. And so it's not ideal. So I try not to eat. Anyway, that is 616, 61 Fahrenheit, 16 Celsius, microbe.tv/TWiV. TWiV at microbe.tv is where your questions and comments go. But who's listening now anyway, right? microbe.tv/contribute if you would like to support our work here at TWiV and other podcasts. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, and once again, I find myself just brimming over with gratitude for this whole thing. No joke. This is wonderful. Thank you.
I have gratitude for all of you participating. I'm amazed that I don't end up alone sometimes because we're doing a lot of these. I really appreciate it.
I just can't get enough of it. It's amazing. I'm thinking about this thing all day long, and I don't get tired of it.
I enjoy our conversations. Yeah. It's a lot of fun.
Yeah.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.